The epigenome is the complete description of all chemical modifications to DNA and histone proteins that regulate the expression of genes within the genome. It is an essential element for the establishment and maintenance of cellular identity. Recently, our understanding of the epigenome has been growing due to the development of tools by which we can edit the epigenome in a locus-specific manner. In particular, Zhu and colleagues have revolutionized our ability to edit the epigenome by being the first to create a synthetic, programmable histone deacetylase using an adapted version of the CRISPR-Cas9 system. The synthetic HDAC by Zhu and colleagues consists of three components: a deactivated Cas9 nuclease, a full-length human HDAC3 fused to the deactivated Cas9, and a guide RNA to target Dcas9 HDAC3 to a specific locus within the genome. The researchers hypothesized that targeting Dcas9 HDAC3 to endogenous promoters could induce histone deacetylation and alter gene expression. They chose to test the effect of Dcas9 HDAC3 on three distinct genes in the murine neuroblastoma cell line N2A, including SMN1, MECP2, and ISO1. These genes vary in expression level as measured by RNA-seq and RT-PCR, and they vary in histone acetylation states of H3K27 at their promoters, as measured by chromatin immunoprecipitation and ChIP-seq. The different expression levels and histone acetylation states of these genes allows the researchers to test their Dcas9 HDAC3 in a variety of contexts. The researchers designed several gRNAs complementary to different target sites within each gene's promoter, allowing them to determine the ideal position for HDAC-dependent regulation of gene expression. The researchers cloned the Dcas9 HDAC3 and individual gRNAs into lentiviral plasmid and transiently co-transfected N2A cells. As controls, they used a mutant Dcas9 HDAC3 with a single inactivating mutation within HDAC3's catalytic domain and a non-targeting scrambled gRNA. The results of Dcas9 HDAC3 activity on gene expression of SMN1, MECP2, and ISL1 genes provides a comprehensive picture of Dcas9 HDAC3 function. The effect on gene expression was measured by quantifying mRNA levels of each gene with RT-PCR relative to mRNA levels in the mutant Dcas9 HDAC3 cells. In all genes, significant reduction in gene expression was only observed when Dcas9 HDAC3 was targeted by gRNA to a region of low H3K27 acetylation within the promoter. This includes gRNA3 for SMN1 and MECP2. The same effect was not observed when mutant Dcas9 HDAC3 was targeted to the same locus, indicating the repressive activity requires a functional HDAC3. None of the gRNAs targeting Dcas9 HDAC3 to regions of high H3K27 acetylation, including gRNA 1 and 2 in SMN1 and all gRNAs in ISL1, lead to a significant change in gene expression. Interestingly, reduction of gene expression was observed when Dcas9 HDAC3 was targeted to regions of high H3K27 acetylation downstream the transcription start site with gRNA1 and gRNA2 in MECP2. However, in this case, the effect on gene expression was nonspecific and is likely independent of HDAC3's enzymatic activity, but rather due to steric inhibition of transcription when Dcas9 HDAC3 binds downstream the transcription start site. Lastly, it can also be observed that there is no significant reduction in gene expression when Dcas9 HDAC3 is targeted to a region of low H3K27 acetylation with gRNA4 and MECP2. This indicates Dcas9 HDAC3's limited range of efficacy when bound to a region that extends 101 base pairs beyond a region of high H3K27 acetylation. Overall, these results confirm Dcas9 HDAC3's ability to repress gene transcription when targeted to a location at or near the tail end of regions with high H3K27 acetylation within a gene's promoter. This provides a positioning guideline when designing gRNA target sites in future studies. Next, the researchers wanted to identify the effect of gRNA dosage on the degree of H3K27 acetylation at the MECP2 promoter. Initially, Zhao and colleagues developed MC3T3E1 clonal cell lines which stably express Dcas9 HDAC3 and gRNA GFP. They chose three specific clonal cell lines which express relatively uniform levels of Dcas9 HDAC3 but different levels of GFP representative of gRNA expression. Then they measured the level of H3K27 acetylation in the chosen clones through chromatin immunoprecipitation followed by quantitative PCR. The results showed significant loss of H3K27 acetylation in genomic regions immediately adjacent to the gRNA targeted sites in both clones 1 and clone 2, but in clone 3, expressing low gRNA levels, the level of H3K27 acetylation is unaltered compared to the mutant clone. Overall, this suggests that a minimum level of gRNA expression is needed to achieve H3K27 deacetylation at this locus. Additionally, by performing ChIP-seq on the mutant clones as well as clone 1 and 2, the researchers could compare the level of H3K27 acetylation surrounding the transcription start site of MECP2. This led them to observe a decrease in acetylation across the MECP2 promoter in clone 1 and 2 relative to the mutant clones, and found that this reduction spanned the entire peak of H3K27 acetylation observed in mutant clones. These results demonstrate the range of DCAS9-HDAC3 deacetylation around its binding site. Next, the researchers examined the effect of H3K27 deacetylation on MECP2 and MC3T3E1 cells. It is shown that MECP2 was significantly upregulated, suggesting H3K27 deacetylation promotes gene expression. However, in N2A cells, DCAS9-HDAC3 deacetylation has a repressive function on MECP2. The opposing effects on MECP2 observed in these two cell types suggest HDAC3's ability to both activate or repress transcription of a certain gene. Lastly, the researchers identified the off-target activity of DCAS9-HDAC3. They compared H3K27 ChIP-seq profiles of clone 1, clone 2, and mutant clones at loci harboring predicted gRNA binding sites with up to 4 mismatches. The results show no significant difference in the H3K27 acetylation of the mutant clone versus clone 1 and 2 across all loci, indicating minimal off-target H3K27 deacetylation. So what are the key findings of this study? DCAS9-HDAC3 can function as a synthetic histone deacetylase in a locus-specific manner. It is guided to its target locus in the genome by a single guide RNA, which is complementary to the target sequence. For optimal DCAS9-HDAC3 activity, the gRNA needs to be targeted to a region adjacent to H3K27 acetylation enrichment within the promoter region of a gene. Once bound, HDAC3 can catalyze the H3K27 deacetylation at a distance of 1 kb beyond its immediate binding site. The level of acetylation is dependent on the gRNA dosage the cell is transfected with. The final outcome of H3K27 deacetylation by DCAS9-HDAC3 is either the activation or the repression of gene expression. Further work needs to be carried out to address the function of DCAS9-HDAC3 in a broader context. It needs to be addressed whether DCAS9-HDAC3 can function at other promoters and in other cell types, alter gene expression when positioned at other regulatory elements such as insulators or enhancers, modify other histone marks besides H3K27, and lastly, it also needs to be determined by what precise mechanisms HDAC3 deacetylation leads to gene repression or activation. Nevertheless, DCAS9-HDAC3 can be added to our toolbox of epigenome editing techniques. We have many questions about our epigenome that remain to be elucidated. How exactly do epigenetic features lead to specific biological phenotypes? What are the precise mechanisms by which epigenetic marks get inherited across generations? How can we exploit our understanding of the epigenome to treat complex diseases? Perhaps now we can start to more effectively uncover the answers to these unsolved mysteries.